Last time, children, I had talked about transforming principle and its biochemical characterization. So we move ahead with the fourth class today, or fourth lecture today. In this, we will talk about Hershey and Chase's experiment to prove that DNA is the genetic material, the final proof. DNA as genetic material, experimental proof. Alfred Hershey and Martha Chase collectively carried out the entire process, the entire steps to prove it. For it, they used the bacteriophages. Now, what are bacteriophages? Bacteriophages are viruses which infect bacteria. At this point, it is important for us to understand what are bacteriophages, how do they actually replicate. Now, bacteriophages, these are viruses. These are viruses which infect bacteria. Exclusively, they infect only bacteria. Now, these bacteriophages, you might be recalling, we had learned earlier that these bacteriophages, its shape is somewhat this way. Okay, now these bacteriophages, they have the a component. Okay, they have a component inside its shell, so to say. That means the covering, inside the covering. So basically they are made up of, when we try to find out the component that makes up its body, it was found that only two components make up the entire particle, the virus particle, that is protein and DNA. It is only made up of protein and DNA. Only these two components make up its body, or make its whole structure. Now, when they infect, now how do they propagate or how do they grow? What happens is they will infect. Now these bacteriophages, they infect bacteria. While during that infection process, what happens? It was observed that one part, not both, one of the elements, either elements means one of the components, either protein or DNA maybe, it enters inside the bacteria and starts propagating. That means inside the bacteria, it starts producing many copies. It produces many copies. So one part of it, either protein or DNA, enters into the body and one part remains behind. That means outside. So whatever enters inside, whether it be protein or DNA, whatever enters inside, that is capable of replication. So genetic materials are capable of replication. So what their search was, Hershey and Chase's search was that to detect what is entering into bacteria. Whatever is entering into bacteria, that is the genetic material. It was confirmed. So this was the principle based on which they carried the steps for this particular proof, to prove it. Okay, now we will see how they did it. They used radio-labeled phosphorus P32 isotopic form and radio-labeled sulfur S35 isotopic form. Why did they use these two elements? Because, as it was already known, it can be either protein or DNA that must have entered the body of bacteria, the cell of bacteria. Proteins, invariably, they will contain sulfur. Okay, and the DNA contains phosphorus. So what was done? First step was in preparing the bacteriophage. Two types of bacteriophages were prepared. One was radio-labeled phosphorus. One was with radio-labeled phosphorus. The other group was with radio-labeled sulfur. That means they allowed the bacteriophage to grow in two batches. They allowed them to grow in one what was provided? Radio level phosphorus. In the other batch, radio level sulfur was provided. So the batch the first batch which was growing in radio level phosphorus will have all the DNA component in its body, okay, in it. That means whatever number it replicates into, all of them will be radio labeled phosphorus. And in the other one, it will be all the protein, all of them will be with radio labeled sulfur. The sulfur component will be radio labeled. So this way they got two categories of bacteriophages. DNA with 32 phosphorus, protein with 35 S. Then what they did? After this, they allowed for infection. Okay, they allowed for infection. When they allowed for infection, the bacteriophages were allowed, that means the viruses were allowed to infect on the bacteria Escherichia coli. Infection started, okay, they infected. The first step, infection, where the bacteriophages infected Escherichia coli. During that infection process, what will happen? One part of the bacteriophage, it might be the DNA or it might be the protein, entered into Escherichia coli. That was certain. Then they allowed blending. That those Escherichia coli, they allowed, that means agitation. Okay, vigorous agitation was allowed following the growth of Escherichia coli. Escherichia coli will, inside the Escherichia coli, definitely bacteriophages have grown. Now that content was subjected to vigorous agitation, after which they were centrifuged. During that agitation process, what will happen is there will be separation of the bacteria and the content that is left behind by the bacteriophage. Bacteriophage has left behind something, has left behind either DNA or protein outside the bacterial cell, and something has entered. So this agitation will separate these two. They will separate the one, that is the part that has been left behind. Following which centrifugation was carried out. Centrifugation will also involve vigorous agitation. In this process, what will happen during centrifugation, what will happen is the denser particles will be and the less dense particle will separate out. So thereby what will be separated out? The supernatant and a portion, okay, which will settle down. The portion which will settle down will be definitely heavier one, that is which is containing the bacterial cells, and the supernatant will be made up of the portion that was left behind by the bacteriophages. Now the experiment. So first, for testing proteins. So what was done? Protein coats were that the protein coat was radio labeled, that means as sulfur. Okay, so we assume that out of the two batches, one batch the protein coats have been radio labeled. When they were infected, okay, when the bacteria were infected, then no radioactivity enters the cell. That means after infection, when radioactivity was tried to detect out, then it was found that the cells were without any radioactivity. Experiment 2, the phases where the DNA was radio labeled, that means it contained the phosphorus, radio level phosphorus P32. This one was containing S35. P32 cells that were there. So this the P32 cells, when they infected, now what happened? It was seen that the bacteria were infected. In this case also they were infected, okay, but radioactivity was detected from inside the cells. Then the next step, what was done? The phases grown with radioactive sulfur. Okay, the phases grown with radioactive sulfur, centrifugation, radioactivity in the supernatant was found after centrifugation. Whereas in the other case, when the phases were grown with radioactive phosphorus, subjected to centrifugation, the radioactivity was found in the pellet, the portion which suspended below. The portion which which were settled below was found as a sediment. It was confirmed that it is made up of the Escherichia coli cells. So radioactivity was emitting out from the cells. from the E. coli cells. The supernatant was not emitting any radioactivity. In this case, what happened? The radioactivity is present in the DNA. There is no radioactivity in the surface. DNA means the phosphorus is radiolabeled. So it proves, okay, it proves that radioactivity is being emitted from the portion in the pellet, that is the portion which was found as a sediment below. That portion has the bacteria. So radioactivity is emitting from the bacteria. It means that DNA had entered the bacteria. So it proves that DNA is the genetic material. Now the other case, the more like the it would prove, okay, it will be more confirmed with this when we see this. That is, now even here the infection process in the same manner. Something has entered, one portion of bacteriophage has entered the cell, one portion of bacteriophage has entered the cell, one portion has left behind. Now when it was centrifuged and then it was taken out, the in centrifuge tubes, when the whole solution was taken out, the supernatant showed radioactivity, but the pellet did not show radioactivity. The pellet has the bacterial cells, supernatant does not have the bacterial cell. So the portion which does not have the bacterial cell is emitting radioactivity, means the sulfur is left behind. It never entered the bacteria, and sulfur is a component of protein, so protein was left outside. So what entered inside was the DNA. So in this manner, it was proved that DNA is the genetic material. So proteins are not genetic material, DNA is the genetic material. Thus, it was proved. Then the conclusion, okay, which was arrived at, that only phosphorus 32 found in bacterial cells, sulfur 35 was found in the surrounding medium, indicating DNA enters and not protein, proves DNA is the genetic material which passes from virus to bacteria and not protein. So final conclusion was this, and till now we accept it. Then what are the characteristics that should be present in a genetic material? The characteristics that should be present in a genetic material, it should be capable of replicating. That means it should be capable of producing its own copies. It should be capable of producing its own copies, multiplying on its own. It should be chemically and structurally stable. It should be stable, that means not easily liable to break down. It should undergo slow mutations. It should express in the form of Mendelian characters. So these are the characteristics of a genetic material. Should be able to replicate. Now, out of the three components, protein, RNA and DNA, it has been seen that DNA and RNA both are capable of replicating. Protein is never capable of, cannot produce on its own, cannot replicate similar copy. It can be formed every time a new one has to be formed, but it cannot replicate. It cannot be exact copy of itself on its own. So they do not fulfill the first criteria. Proteins do not fulfill. RNA and DNA to some extent they fulfill the criteria. Chemically and structurally stable. When we look into RNA, it is found that RNA is comparatively unstable than DNA. Why? Because DNA has one oxygen less. Its oxygen in the second position is missing, due to which it will not react easily with any other compound it comes across. Whereas RNA has that additional oxygen, or has that oxygen in a second position, which is always free. It is not bonded to any other compound or any other molecule. As such, they can easily react with some other component and undergo a change quite rapidly. So in this context, DNA was found to be, or is found to be, structurally more stable, chemically more stable than RNA. Then they should be capable of undergoing slow mutations. Both RNA and DNA are capable of undergoing mutations. Rather, RNA undergoes mutations at a more faster rate. Why? Because they are already unstable. So changes can occur very fast in RNA. Whereas DNA, even if change occurs, it will be very, very slow. Then expressed in the form of Mendelian characters, that means the whatever is acting as the genetic material should be able to form proteins, which are the ultimate expression of characters. And DNA can definitely help in formation of the proteins. Even RNA can help in formation of proteins. So both RNA and DNA fulfills these three criteria are fulfilled by both of them, both DNA and RNA, whereas with regard to stability, RNA is found to be less stable than DNA. So, as such, DNA is the genetic material in most of the higher organisms, RNA being the genetic material only in viruses. Otherwise, it is DNA that is the genetic material. The reason being due to the stable nature of DNA. Thank you for this patient listening to this lecture. So in the next one, I will be explaining about DNA replication. Thank you.